Hello, welcome to our poster preview video for Photonics West 2021, titled Multi-Species High-Resolution Mid-Infrared Spectroscopy via Fiber Delivery. Chromacity manufacture a range of light sources, from the fixed wavelength ultra-fast lasers at 520 and 1040 nanometers, to OPOs with integrated pump and output in the near IR and the mid IR. The work in this presentation is using the idler output of the near IR OPO, around 3.3 microns, which has an average power up to 350 milliwatts. The Chromacity light sources can be used for a variety of applications, but this poster presentation concentrates on IR spectroscopy for remote sensing using the near IR OPO. The Chromacity near IR OPO is compact, air-cooled, and an affordable source for IR spectroscopy. The good beam quality allows for long path lengths and for compatibility with high-resolution FTIR spectrometers. The high brightness again allows for long path length and also for high sensitivity measurements. And the broad bandwidth means that you can probe multiple species simultaneously. It's also tunable from 1.4 to 4 microns, meaning many fundamental vibrational bands can be accessed. These points make it well suited to applications such as atmospheric sensing, which often require remote delivery over open paths up to kilometers in length, or fiber delivery to more inaccessible environments such as pipelines or reaction vessels. It is this second point that will be the focus of the poster presentation today. In this work, we explore a flexible, easy-to-use technique which can be used to conduct remote, fiber-delivered multi-species spectroscopy in a spectroscopically cluttered landscape, containing both narrow and broad absorption features. The Chromacity OPO light is routed through a Michelson interferometer before launching into fiber. The output of the fiber enters the environment of interest, here simulated by a 10.5 meter Herriot cell connected to a closed-loop air delivery system. A detector at the cell output measures the spectrum. A reference spectrum is also recorded before the light enters the fiber. The fiber type used here is zirconium fluoride, but could be a range of fibers, including hollow core fiber delivery systems. In this example, we show quantitative, high-resolution, simultaneous detection of water, methane, methanol, and MEA. The detection of these chemicals demonstrates monitoring of multiple hazardous chemicals in enclosed environments for health and safety reasons. Once a spectrum has been recorded, our fitting algorithm determines the concentration of the species present. This is done using a global optimization in which the illumination spectrum is modeled as a many-point spline and co-optimized with database spectra of participating species to obtain their concentrations. For species with line-like absorptions, this means baseline correction can be done without the need for a reference spectrum. An example is shown in panel B of the figure. This is a significant benefit for cases such as atmospheric sensing, where it is difficult or impossible to measure a reference spectrum without the species of interest. For improved accuracy, in the case where species have broad absorption features, the ratio of the reference detector spectrum measured before the fiber to the fitted envelope of a cell containing only ambient air constitutes a transfer function. Therefore, for subsequent measurements in which additional chemical species are present, the spectral envelope can be retrieved by multiplying the reference detector spectrum by this transfer function. Examples are shown in A and C of the figure. In more detail, part A, or panel A of the figure, shows a cell containing MEA, methanol, water, and methane. The main plot shows the illumination envelope as a dotted line, the measured spectrum as red, and the best fit as blue. The small panels above show the relative contributions of each of the absorbing species, and the bottom panel shows the residual. The detection sensitivity of any of the chemical species fitted can be estimated from noise equivalent absorption analysis. This is shown for MEA in the top row and methane in the bottom row. These are measured from a cell containing only MEA and ambient air, that's column 1, or panels A and C, and from a cell containing MEA plus methanol and ambient air, that's column 2, panels B and D. For a cell containing only MEA and ambient air, so panels A and C, the detection limits of 290 and 890 parts per billion are obtained for methane and MEA respectively, using this setup and averaging 64 spectra. The panels in the image show the variation of this detection sensitivity across the spectrum. So where the species absorbs strongly, you get better detection sensitivity. Hence the shape of the plots following the absorption spectra of the species. The impact of an additional species with a complex spectrum, in this case methanol, on the detection sensitivity of the others, MEA and methane, is explored in column 2. You can see that the detection sensitivity is reduced by the presence of the complex spectrum, but not to an overwhelming degree. The detection sensitivity can be improved using longer interaction path lengths, so longer cells, and by averaging more. The previous slides showed data which were the average of many spectra. However, individual spectra can also be fitted to give time-resolved concentration measurements, as shown in column 1. This also allows the precision of the measurements to be estimated by calculating the Allen deviation, shown in column 2. The Allen deviation shows the expected improvement with averaging for species with stable concentration. For example, methane shown in the top row here. After 16 measurements, we see that the precision of the methane measurement is better than 20 parts per billion. For species in which the concentration changes, such as MEA, then you can track the change in concentration over time. In the experiment here, the samples are taken at 20-second intervals. However, with this type of setup, we have shown operation at sampling rates of up to 3 seconds per spectrum. Provided the concentrations and experimental setup are stable, then the precision can be improved with more averaging. In addition to the fibre delivery work shown on the poster in the previous slides, I want to show an example of this technique being used for remote standoff environmental monitoring, in this case hydrocarbon detection. So again, the Chromacity OPO light is coupled into a Michelson interferometer before sending to a distant target. The light scattered back is collected by a telescope to give the spectrum, and this includes absorption features from any gases present in the light path. It's demonstrated for simultaneous methane and ethane quantification, but is applicable to all molecules with absorptions in the wavelength range. For example, carbon dioxide, nitrogen dioxide, etc. The example shown in the figure and the cited paper there is for a range of 70 meters with backscattering just from a rough foil target, but is applicable to kilometer lengths with a retroreflector. And it was shown to have a methane detection precision as estimated from the Allen deviation of 40 parts per billion for 5 minutes of averaging. So in conclusion, the Chromacity near IR OPO is a high-performance, affordable light source for multi-species IR spectroscopy. Our fitting algorithm enables quantitative measurement of chemical concentrations, even for species with broad continuum-like absorption features. Remote delivery of the light is easily implemented, either via fibre or over open paths. The detection sensitivity and precision can meet relevant environmental monitoring specifications. Thank you for listening to this Photonics West poster preview video. If you would like any more information on this technique or any of the Chromacity products, please visit the website or get in touch with the contact details shown. Thank you.